In this lesson, we're going to be talking about something called a test cross. And this is really just a continuation of what we were talking about in the last lesson, the monohybrid cross. This is something that is used, it's a monohybrid cross, but it's used for a specific purpose. So if you have an organism that is displaying the dominant phenotype, you can't tell just by looking at it whether it's homozygous dominant or heterozygous. We can't figure out the genotype just by looking at something when they display the dominant characteristic. So, for example, a tall plant could be capital T, the homozygous, or capital T, small t, heterozygous. However, if you have an organism that displays the recessive phenotype, then you know the genotype because there's only one option. It has to be homozygous recessive. That's the only way that that trait is expressed. So short plants are always small t. There's no other option. So if you have individuals displaying the dominant traits and you want to figure out what their genotype is, you do something called a test cross. So when you do a test cross, you take the individual organism that has this unknown genotype and is displaying the dominant trait, and you cross it with a homozygous recessive individual. So then you look at the phenotype of the offspring, and that will allow you to figure out what the genotype of the unknown parent was. So when you look at the offspring, if the individual of unknown genotype is homozygous dominant, then all of the offspring are going to be dominant. There's no way they will be able to express the recessive trait because they won't be able to get a recessive allele from that one parent. However, if any offspring at all do display the recessive trait, then you know that the individual of unknown genotype must be heterozygous because they were able to create offspring displaying the recessive trait. And the key here, though, is that you look at a lot of offspring. Okay, you can't just look at one and say, oh, it's dominant, therefore it must be homozygous, right? You need a lot of offspring to make a reliable conclusion in a test cross. So if we look at an example, in guinea pigs, black fur color is dominant over white fur color. So how do we determine the genotype of a black guinea pig? So the answer is we perform a test cross, which means we're going to take a black guinea pig and cross it with a white guinea pig and see what happens. So for the mystery pig, there are two possible genotypes, right? It could be homozygous dominant, so big B, or it could be heterozygous, big B, little b. Either of those create a black guinea pig. The white guinea pig must be small b. If we look at what happens if we put this info into Punnett squares, we've got the dominant individual with one big B allele, and then we don't know what the other one is, crossed with the white guinea pig, which is little b. So if we put in the information that we have, right, so we know that the one parent is going to give a small b allele, and the other one will give one big B and then something else. And then if we start to fill in the Punnett square, we see that at least 50% of the offspring are going to have to show the dominant trait. But we don't know about the other 50% because that depends on that missing allele. So we have those two possibilities. So if we look at the one where the mystery pig is homozygous, right, and we put in that capital B, we find that all of the offspring have to be black. That's the only option. There is no possible way to get a white offspring if the black pig is homozygous. Whereas if the black pig is heterozygous, half of the offspring would be expected to be white. But the reality is, as long as you get one, then you know that that allele had to be there. So key points for test crosses. The organism with the dominant trait is always crossed with an organism with the recessive trait. That's the only way you get your to figure out what the genotype is. If any offspring show the recessive trait, the unknown genotype is heterozygous. If all the offspring show the dominant trait, the unknown genotype is likely homozygous dominant. But notice how we have likely there because, say, you have 50 offspring, it's likely it's homozygous dominant, but you just can't be sure that that 51st one wouldn't have been recessive. Chances are it wouldn't, but there's some probability issues happening there. And so that's where the fourth point comes in. You need large numbers of offspring to get reliable results. So those are test crosses.